Okay, might be we can get started. So,
Okay.
Devra, Vipin, go ahead. Yeah.
Yeah. Hello, everyone. Good morning. So today I'm presenting our paper Forte, which has been accepted to ICLR 2025. Forte stands for Finding Outliers with Representation Typicality Estimation. And this is a paper on how you can do good out of distribution detection. So this talk is going to be structured around first exploring what out of distribution is, why it is important, and I'm going to um, set up some of the theoretical results that unsupervised out of distribution detection relies on, explain how some of the relevant techniques work, and finally, I'm going to introduce our technique results and strong results in domain generalization. And in the final few minutes, we're going to try to very quickly show how people inside Icicle can use Forte in their own projects. Okay, so let's start with OOD, what and why. So if you've seen this very funny episode from the TV series Silicon Valley, there is a character Jin Yang who is tasked with building a model that can detect food that you have on your plate. So what kind of food is it? And what he ends up having a problem with is that he has got very little data, and he ends up only being able to build a classifier that can detect whether it's a hot dog or not a hot dog. And this is really the problem a lot of us face as ML practitioners, right? We have limited data, and the data that we train on is usually not the only data that we see when we actually put this model into the real world. So let's say you're building a very simple cat versus dog classifier. You can obviously get things from your test distribution of either cat images or dog images. But what you might end up also getting is a photo of a lion, which would be a near out of distribution data. So the difference between near OOD and far OOD mainly relies on the semantic content of these images and how dissimilar they are from your individual, the images that you trained on. So a car that is shaped like a cat might be a far out of distribution data point. So moving beyond funny things, let's formally try to define it. So you might have a distribution shift is when the training data is different from the test time data. So you can have covariate shift where your P of X changes and P of Y given X remains constant. So a good example would be in, let's say, a spam filter. You have a training data that can consist of formal business type emails, but in deployment, you encounter personal emails. And these personal emails may have different vocabulary, structure, etc. So the input distribution, that is the email content, has changed, but the relationships between the input and the labels and like what constitutes spam remains the same. You can have label shift. Let's say in a medical diagnosis setting, you have a model that's trained on a population where only 5% of the people have that certain disease. But when you deploy it, let's say in a specialized clinic, which only caters to that type of disease, maybe 30% of your patients have that. So the prior probability of the classes then changes, but the features of the disease remain constant. Um, okay, next slide. Concept shift. So concept shift happens when, let's say, you have trained a sentiment analysis model based on a movie review or something like that, right? And you want to use that model that has been trained on movie reviews on analyzing, let's say, e-commerce reviews, right? So whereas the word unpredictable might be a good thing for movies because it indicates that it has an exciting plot or something like that, it is a negative for products, because this would mean that they have unreliable performance. So the relationship between the features and the labels changes when you have concept shift. And finally, distribution shift is something like general distribution shift can be you have a self-driving car trained in California, and you deploy that in like very snowy Cleveland, where your input features, that is the road visibility or like your surface texture, all of these change, and the frequency of the scenarios change. You can have more slippery condition, and the relationships between input and like the safe driving behavior might also change. So you might want to maintain larger braking distances, etc. So these are real problems that you face day to day in ML systems. But you can simply express it as such, right? You have a training in distribution data that is this nice Gaussian that you train from, right? And at test time, you get a mixture distribution, which is mixed with this parameter alpha, which dictates how much of your original distribution is in your data and how much of your OOD data is actually inside that too. So this part is your out of distribution data that you want to identify. So this is actually a non-negotiable problem for safe and deployable ML, because most of our models, as we've talked about before, are bounded by the knowledge they possess based on the data that they've been trained on. And when we go beyond this bound, we get silent failures where your model might give bad predictions with good confidence. And we want to allow some amount of safe fallback, where if it does fail, a human can interfere or it does not give wrong decisions. So let's talk about what is wrong with some of the current out of distribution detection methods. So a lot of these methods require class labels. Labels are expensive, scarce, and often incomplete. Some OOD methods require exposure to OOD data during training. There is a paradigm called as outlier exposure, where methods would need to see these outliers during training. And in reality, this is an impossible paradox, because how do you train on unknown unknowns? So that's a conceptual problem with some OD techniques. You might have restrictions on the architecture of the models, whether predictive or generative. So a good example of this is density of states estimation that we're going to talk about right after this. And a lot of these techniques actually have post hoc methods where they do things like temp scaling, like, you take the logits and you manipulate these logits, a lot of them very, very in sophisticated manners. And this would add a lot of latency to any applications, and this is very cost prohibitive for any large scale deployment scenarios. Finally, none of these methods present very strong domain generalization. So by strong domain generalization, I mean that most of these methods operate on top of the logits of your neural network. And therefore, because your neural network has not seen a lot of data, it cannot generalize to beyond the very specific distributions that, or the associated distributions near it, that it was trained on. So what we try to fix is create a generalized and data-centric approach. I'm going to now present some of the theoretical foundations behind what makes Forte work, just to give you an intuition for why it works. And really the reason why I'm doing this is because Forte is a very simple technique, as you'll figure out in the later part of this talk. And a lot of people struggle to grasp why it's working. So I'm going to just give you some, you know, uh, background on why it does work. So one theorem, we're going to cover three theorems mostly. One is the Gaussian annulus theorem. The Gaussian annulus theorem is a fundamental result in high dimensional probability. So in very simple terms, when we sample from a high dimensional space, like from a high dimensional Gaussian vectors, their lengths tend to concentrate around a narrow annulus or ring with a radius of square root of n. So over here, what it says is that let X be a random vector in Rn, sampled from a standard Gaussian distribution. I of n is the n by n identity matrix. Then for any small epsilon greater than 0, the Euclidean norm, that is the distance between the origin to the points itself, concentrates around this square root of n. So to just give you an idea, if you draw these points from a two-dimensional distribution, you see that um, between 0.9 of root n and 1.1 of root n, in two-dimensional setting, you have 14.4% of the points. And as you increase the dimensionality, you start seeing that more and more points start falling within this shell. And this is your the orange dotted line is your expected distance, and the green one is your observed distance, right over here. So as we tend towards like a 1,000-dimensional setup, we see that almost 100% of the points fall within this little shell in 0.1, 0.9 of root n and 1.1 of root n. The second theorem that we want to discuss is the concept of typicality, which is very common in information theory. So in very simple terms, what typicality means is that a typical set is a set of sequences whose the empirical probability is close to the theoretical entropy of the source. So it's used in a lot of compression and coding theory work in information theory. So typical sequences have a probability very close to 2 to the power of minus n h of x. And h of x is the entropy of the source. So despite being a small subset of all possible sequences, the typical set contains almost all the probability mass as the n grows larger. And this brings us to AEP, which means that the probability that a sequence x to the power of n belongs to an epsilon typical set reaches approximately 1 as n approaches infinity. So for a sufficiently large n, almost all sequences fall into the typical set, meaning that their empirical entropy will match the true entropy of the source. Randomly generated sequences, this will mean that they will have more predictable properties in higher dimensions. So let me show you what this means, right? So let's say you have two distributions. One is your in distribution, one is out of distribution. These are very simple Gaussian. So if you have a two-dimensional distribution, you will see that you expect it to fall close to the root n expected distance, as we saw from the Gaussian annulus theorem. But in two dimensions, they overlap against each other. But when you do move towards higher dimensions, you can tell what distribution you're drawing from just from the typicality. So again, as you move towards the 1,000-dimensional setup, your atypical samples, that is your out of distribution samples, move further away compared to your in distribution samples right over here. And what you see is that your AUROC eventually, this is on log scale over here. So your AUROC, as higher dimensions come, you tend towards a perfect classifier. Finally, the third theorem we're going to talk about is really this concept of density of states. So let's say this is used pretty often in statistical physics or a lot of undergrad physics. So if you have a physical system consisting of a set of particles or a data set consisting of a certain number of data points, for each particle x, you could compute a scalar property This could be the energy, log probability, max coordinates. Max coordinates is L infinity norm, right? Etc. And the goal is to determine whether a particle is atypical just on the value of the scalar property. And this will be done if it is if something is atypical, that means it is inconsistent with the equilibrium distribution of the system. So this really means that it measures the density of state measures how many different ways particles can arrange themselves while producing the same measurable outcome. So you're integrating over all your possible states that yield the property T. And this is related to entropy in thermodynamics. So think of it like counting all the ways in which you can roll a specific sum with dice, and your density of states tells you how many combinations produce that particular result. So with concentration of measures, what this property says, as the system dimension n approaches infinity, the distribution of the property, the T of n, which is the scalar property that we calculated, under the equilibrium measure, concentrates sharply around the typical values of T. So let me just show you what that looks like in using the graphs that we were looking at earlier. So if you calculate three different metrics over here, so we're calculating log probabilities, we're calculating energies, and max components, right? What we see is, again, at a lower dimension, um, the the dashed line over here actually is the typicality score. I want to emphasize. The blue one is the typical density of states, and the red one over here is the atypical density of states. So this typicality score should match up with your blue line over here, which it does not at lower dimensions. But as you go towards higher dimensions, just looking at what these density values look like and what these energy and max component values will look like, you can tell whether something is in distribution or not. The reason we want to do this is because if we operate over very high dimensional spaces, looking at high dimensional embeddings might not be enough because there is just too much noise in it. But if you look at um, the the distribution of these individual metrics, you end up being able to tell and identify when certain points are drawn from an area of higher typicality versus lower typicality. Over here, again, meaning that you can use these estimates to be able to go towards a perfect classifier at detection. So I promise that's the end of the math part for now. The rest of it is going to be very intuitive. So the key takeaways here being that typicality means that most sequences look typical. That is, their empirical entropy matches the true entropy of the source. The Gaussian annulus theorem says that most Gaussian vectors lie within this very thin shell around the radius of root n. And rare events is that the probability of finding a system in an atypical state, that is like, with a bad density of states, decays exponentially with the increase in the system size. So previous techniques have tried to use typicality estimations in out of distribution detection. The best technique and the most recent one is density of states estimation, called DOSE over here. So like we did compute those previous metrics before, like energy, log probabilities, etc. What density of states estimation does to predict whether a sample is OOD is it calculates these metrics based on generative models. So it will train a bunch of VAs, or like one VA, let's say, on your distribution, your in distribution data, and then it would score whether an in distribution sample is given or an out of distribution sample is given, what these metrics look like. And basically, if you draw if you can fit a one class SVM or a kernel density estimator on top of these values based on their typical results, you can tell when something is atypical and flag that as out of distribution. The issues with this happens though, because you're constrained to using generative models. So let's say you're doing a nightly model release. So if you have a new data set that's changing pretty quickly, what happens when a few samples change? You will have to train a new generative model. Glow models have invertible architecture, so they're not very efficient. They have high memory requirements because they actually do exact log likelihood evaluations. VAs, on the other hand, struggle when you have few examples available. So you can't train VAs if you have, let's say, 1,000 images. It will be very difficult. And this will give you a poorly structured latent space and degraded performance, even if you manage to train a autoencoder. So what does Forte do instead? So in Forte, what we do is we use self-supervised methods instead of generative methods. So we look at Dino V2, CLIP, and VITMSN kind of metrics. These are self-supervised learning methods. These are very commonly used. And I'm going to explain Forte to you from a very geometric standpoint, right? So you have your training data, which is XIR, right? And your test time data is XJG, let's say, right? So let's say your in distribution data forms inside a 2D space is laid out like this. So you can start with one point and you can look at its k nearest neighbors. So let's say over here, you set the kth nearest neighbor to be 3. You can have an NDK of 0.41. Now what we want to do is draw a Euclidean ball over here, right? We want to draw a Euclidean ball centered around the point X with the radius of nearest neighbor distance of K of X. And you want to take the set of all these balls, that is, for every single one of these points, you're drawing balls. And then you consider we're actually computing a local transform based on the these Euclidean balls. So what we see over here, this is just the expression of what we discussed right now. You have the set of all Euclidean balls over here. And we're calculating, instead of the previous metrics, we're calculating these 4 metrics, right? These are the scalar metrics that we compute for density of states estimation. And then we fit a kernel density estimator or whatever on top of what these metrics look like when the training distribution and the testing distribution is the same. So you only train on your in distribution data, you split that into half, you compute these metrics on top of them, and then you end up with a distribution of what these metrics look like. And when you do have the intuition being that when you do have something that is out of distribution, these metrics are going to look very different for an out of distribution sample compared to an in distribution sample. So what do these metrics really mean? If I have to simplify it, your precision is saying, is there a test point? So over here, the reference samples are your in distribution samples. And your test samples are from your mixture distribution. So they can be either from your in distribution sample or your out of distribution sample. So is the test point inside a reference point? And this is a binary statistic. It's 1 if yes, 0 if no. So 1 meaning less OD. Recall, on the other hand, is the fraction of reference points inside each test point ball, right? So this is a continuous metric. This is between 0 and 1. Higher is less OD. Density looks at the fraction of, so this is illustrated by the concentration of the colors over here. This is the fraction of reference balls containing the test point. and we do some normalization on top of this. So higher density means higher chances of typicality, right? And coverage finally is asking whether is the nearest reference point closer than the test ball's radius. So this is also a binary metric where 1 if yes, 0 if no. Let me show you what some of this looks like. So let's say you set k equals 1 over here. You're just looking at the nearest neighbor distances. We see that most of these test points like are inside the reference ball of similar of points close to it. Apart from this one, this point is an outlier because it has a precision of 0. Recall over here, if we look at this point is just outside of its nearest neighbor distance. That is, the distance between this and this is lesser than this point. Therefore, the recall for this is 0.0. The density for this is there are 1, 2, 3, 4, 5, 6, and 3, 4. So I believe there should be 4 by 6 over here, which is 2 by 3, and therefore density of 0.67, right? So it's this point is within 4 of the 6 circles. Therefore, it's 0.67. And then there are coverage being like, is the nearest reference point closer to the test pulse radius? And then it's 0 over here and 1 for these. And this k is actually a hyperparameter that you can adjust. So let's say you want better granularity, you can do something like increase the value of k, and your recall value from 0.00 that now becomes 0.5. We conduct all experiments with a value of k equals 5. So Forte, as I said, is very, very simple, right? The key steps are as follows. You extract the features using clip, VITMS, and Dino V2, etc. You compute the per point metrics that we just discussed. And then you train a density estimation model based on these metrics, right? And let's say you only have precision per point, and these are your in distribution samples, and these can be your OOD features. So this is your decision boundary based on just the precision per point metric, right? This is a simplified representation, which is why there are a bunch of OOD features that actually make their way into the in distribution space. But this is just to give you an idea that we're trying to estimate the manifold of the original data as closely as possible with the help of these transforms. So to just illustrate, this is what the 4 metrics look like under no shift. So your blue one is the in distribution data, and your pink one is OOD. Why this is magenta is because when there is no shift, these distributions are lined perfectly on top of each other. So therefore, it's this magenta color. But if you just shift it by 0.01, right, this is just a tiny shift that we make to the mean of the distribution, and they start diverging very quickly, as you can see over here. And under a small shift, that is a 0.1 change to your mean and covariance matrix, you end up with very different distribution of these data points. And this is what we're really looking at. So let's talk a little bit about benchmarks and what how we perform using this technique. So this is comparing Forte against supervised out of distribution techniques. So supervised OOD techniques use labeled OOD examples alongside in distribution data. The models explicitly learn how to distinguish between in distribution and various OOD classes. And this is often formulated in a binary or a multi-class classification context, right? Generally, these techniques end up achieving higher AUROCs than unsupervised techniques because it has seen examples of what is not in distribution, and but this requires collecting representative OOD samples that can be challenging, right? So in all of these graphs, you'll see, these are all the publications and like, the methods, all ICML, neurips, etc. And this is how, the state of the art has evolved over time, right? And you can see in all of these, our technique outperforms everything else, either by a large margin or even if not by a large margin, small margin, but outperforms all other known techniques over time. This is reporting the AUROCs. Unsupervised OOD detection only uses in distribution data, so no OOD examples are available. The model must, infer what constitutes OOD based solely on the characteristics of the in distribution data. I want to emphasize that that Forte is an unsupervised technique which outperforms known supervised techniques. So typically in unsupervised techniques, you rely on density estimation, reconstruction error, or confidence scores. And as you can see, even on a task like CIFAR 10 versus 100, density of states estimation was the prior state of the art, which was like the unsupervised SOTA for at least 2 or 3 years now, until we outperformed it by about 40 points, 0.4 AUROC, which is a big jump. And again, with everything else, we'll be outperforming. Finally, I want to talk about domain generalization. So outperforming all known benchmarks is good, but we want to actually build something that's usable in the real world, right? So one particular example we picked out of a task was identifying synthetic data samples. So over here, these are images of golden retrievers, actually. And over here, these are the original images on the top row of of golden retrievers. And then latent diffusion models have this image to image setting, which is you can take an image as a prior and then you perform diffusion on top of it to reconstruct an image. And you can change the strength of your image generation, so of the diffusion process. So it adds more noise to it, and therefore it can end up like, creating images that are further off from the original data. So these are your, with 0.3 strength, with 0.5 strength, with 0.7, 0.9, 1.2. So as you increase the strength of the diffusion process, what you see is the pictures of these golden retrievers start diverging more and more from the original data that you started off with. This is just to give to simulate how synthetically data distribution drifts occur, or what mode collapse and like generative model failure modes look like. This row over here, I believe. Hold on. So F1 through F, and then G is, these images were captioned. So I'm assuming this was a person with a dog and a stroller, and like, a person with a dog got generated over here. So these are caption generated images, and these are just class name generated images. So these are just, if you ask it to generate golden retrievers, this is what will get produced. And what we end up seeing via this PCA is that real distributions are actually very varied. And as you go towards synthetically generated data, you end up, getting this kind of mode collapse over here, where you're generating only a specific kind of image compared to like, the real distribution, which is way more varied. And as you increase the image to image strength, actually this collapses. So similarly, in the data drift, we check, and you can use Forte to try and understand how much difference there is between these two distributions. So commonly used generative evaluation strategies are FID, FD at infinity, CMMD scores, etc. And you can see that these are not detecting them as two different. Whereas by the time, like, by image to image 0.7, where even the best CMMD score, this is considered the best method there is right now. This is indicating a lot of similarity, but we can tell the difference between the two distributions very well, which is like 94.1 accuracy. So this shows that not only does it work well for the benchmarks, but also for detecting synthetic data. And again, as you can see, these are almost close to perfect numbers. Finally, the other problem that we're going to talk about is identifying MRI protocols. So clinical data is actually very interesting because most hospitals or more clinics will use a specific type of machine to be able to generate that data. And what commonly ends up happening is that you train a model based on that specific data. And because of that reason, your model performs very well on that distribution. But if you take that model and deploy it at another hospital, that model does not perform very well because the distribution of the data has changed, as in, your acquisition parameters over here for MRI, you're talking about, these fast MRI, OAI, these are sampled using these metrics. And depending on whether there is fat suppression on or no, you will have very different data samples. But to a normal, to me, like to a regular person, these look very similar. So how can you detect the difference between them? And as it turns out, if you consider some of these as in distribution and use it to detect against other out of distribution samples from other data sets, we see close to perfect performance. So not only do we have a technique that works well at the benchmarks, but also works even on medical data that it's completely unfamiliar with. And finally, I want to touch upon the topic of deployments on edge in case that's required. So if you want to deploy on the edge or you want to use specialized data, there seems to be strong evidence to support that you can swap out your Dino V2 or clip with your own tiny vision transformer or CNN that can generate encoders. So this can be, let's say, if you're working on a specialized um, form of data, let's say histopathology images, you can use the embeddings from a network you already have trained on enough of that data instead of our clip, etc., if you really want to. And what this shows is that you can swap out the clip, etc., with a tiny vision transformer, and you will still get 99.29 EUROC at CIFAR 10 versus CELAB, for example. So that's good. So the next thing I'm going to talk about, we don't have too much time. I'm going to show very quickly how you can use Forte yourself. Okay, so this is a demo. I'm going to remove all of this. Now, what's interesting about this is that the Forte API, if you want to use it, this is already live on the ICICLE GitHub. So there is a Forte API right now, and all you need to do is just get this file. And we'll deploy it via pip eventually. But right now, all you need to do is just get the Forte API. And this is just around 500 lines of code that implements everything that you will need. So what this looks like is I can just do get Forte. I just get the raw file from that and save it as this Python file. And this is my demo, right? So what I'm doing is very simple over here. I'm going to download a bunch of CIFAR 10 images and CIFAR 100 images, and we're going to end up training this pipeline. And what's very interesting is that you only have to import this one class from this API, so Forte OOD detector, and you initialize that as such. So you initialize the model with your device, your embedding directory, whatever method you want to use in your nearest k. And like sklearn, you can fit your detector on top of your in distribution data points, right? So you have a detector.fit method, and you also have a um, get OOD scores method. So what this will do is it will give you what the scores are given your in distribution data or your out of distribution data. So you pass the different images to it, and it will just give you the OOD scores for it. Finally, to benchmark it, you can also call the evaluate method, in which you give your in distribution samples and your out of distribution samples, and it will run all the evaluations for you. So I'm just gonna run this for you just to show. I have pre-computed the embeddings for this demo, just so that we can go fast. And this is how quickly. So we do 10,000 train images and 2,000 test images from CIFAR 100, right? And it's just gonna calculate stuff and it's gonna benchmark it. So we can see that we reached an AUROC of about 0.95 using a subset of these images. Our eval took 2.7 seconds. We can, what this says is um, per image to infer is around, this is running on device on my Mac Pro. So this takes me around 0.01 second to get inferences from each, from the out of distribution detector, and to calculate images, the embeddings per sample. It takes about 0.1 seconds. So this can be optimized quite a bit more, but this is just to emphasize that this is pretty fast. So if you are sampling at 10 FPS also, you might probably be able to do it at real time. That is about it. I think we have got 10-15 minutes left. So I want to open it up for questions.